for this Welcome everybody. Happy Wednesday. And wow, that was really cool. That's a little countdown. That's new. It's good to see the evolution of the Meet the Scientist program here. So I'm Brian Magierski with Cerebrum DAO. We've been running the Meet the Scientist series for several weeks now. Every 2 weeks or so, we try to feature a new project in the Cerebrum DAO ecosystem. For those not familiar with Cerebrum DAO, we are a global crowd-funded and community-governed organization that funds and facilitates translational research to therapeutic outcomes in the field of brain diseases, with a goal and a mission of ending all brain diseases. So we intend to be around for a long time, and we do intend to accomplish this mission. So we're early in our journey and are glad that you're here. Today's episode is an exciting one for me, as one of our objectives in 2025 is to really start fulfilling the mission of Cerebrum, not only by funding projects that could lead to therapeutic interventions and focusing on translational research and drug development, but really bridging the worlds of the patient communities with the research that we are doing that could lead to outcomes for that community. So we're really eager to engage patient communities and have people whose lives are affected by the brain diseases we're seeking to prevent or cure, to have active involvement and agency and voice in the work that we're doing and the types of projects that we take on and fund here. So today I'm really thrilled because we are going to be bridging the research and patient community together with the drug development on a specific project around ataxias, which is a category of brain conditions that we're seeking to address with Cerebrum. So I am really excited today. We have Peter Groenen, who leads our translational research development here at Cerebrum DAO, and Dr. Julie Greenfield from Ataxia UK, which is a patient community. They're both going to tell you a little bit more about who they are and the work they do, and then we're going to get started. So Peter, do you want to start, and then we'll hand it over to Julie?
Yeah, thank you, Brian, for the introduction. I'm very excited about the session today. It's bridging research from research to a patient. So my name is Peter Groenen, and I'm a translational scientist. I've been working in the pharma and biotech industry for about 25 years. I decided some time ago to go on my own to help other companies, but now with Cerebrum for already almost half a year, helping them also with the research strategy and the translational science strategy. And very much in the beginning already was made familiar with this project that we have been evaluating, and about that later. So very excited to discuss this also with Julie.
Thanks. So hi, so I'm Julie Greenfield. Thank you very much for inviting me here today. So I'm the head of research of Ataxia UK. So my background's in lab science, but that was a long time ago, and I've been working for the charity for Ataxia UK for many years now, where I lead the research activities. So Ataxia UK is a charity sort of provides support to people with ataxia, with a whole range of ataxias, information, support, helpline, events, conferences, all the sort of things that many patient support organisations do. And in addition, we're a research active organisation, so we kind of do a whole range of things there, whereby we have a small funding programme ourselves, but we also do a lot of collaborative work and a lot of promotion of research and supporting companies or academics, depending on the sort of level of need that they have to try and sort of advance research on the ataxias.
Fantastic. Well, thank you both for joining this and sharing your expertise and discussing the exciting project that Cerebrum is embarking on with our SK Channels project. Why don't we get started and let's talk about ataxia itself and the diagnosis. So many people may not be familiar with the condition. So what is ataxia? What are the types of ataxia? And you know, we are, I believe, looking specifically at something called SCA2, and what would you know, clinically or genetically differentiate that from the other types of ataxias. So maybe a lay of the land for people on the condition itself and the variations of it.
Yeah, so there's many different types of ataxia. So you can get ataxia sort of means a lack of coordination of movement and balance. So essentially you can get ataxia symptoms through a variety of ways. So you can even get it as a result of a stroke, or you can get an acquired ataxia. But the ataxias that we tend to focus more on are progressive genetic conditions. And the spinocerebellar ataxias in particular are a group of around 50 different conditions, which are all inherited in a dominant fashion and are neurodegenerative progressive conditions. And the general sort of symptoms that they all have are these problems with mobility, sort of losing, initially having problems with coordination of movements and gait, and eventually many people are not able to walk anymore and have to use mobility aids. Some issues with coordination of upper limbs and sort of all the associated problems that can lead to, slurred speech, and a number of other associated symptoms. So those are sort of the generic symptoms that all ataxias have. And then specifically, when you go and look down at specific ataxias in SCA2, for example, there's a few associated things, such as people can get peripheral neuropathy or some sort of movement of the eyes, tremors, cramps, some additional sort of neurological symptoms. And the issue with slurred speech also later on might also lead to problems with swallowing and sort of, you know, there's various sort of things around quality of life that then get affected through those things. And later on, you can also get some cognitive issues and some dementia in later stages. So those are sort of the symptoms. In terms of the genetics, it's caused by a specific mutations in specific gene, the ataxia 2 gene, and so that's why, you know, it's one specific condition. Although there's many similarities with the other ataxias in terms of the cause, it is a specific cause. And once again, the spinocerebellar ataxias also vary in terms of when symptoms start. So in some cases they can start very early on or later on, and in SCA2 the average age is about 30. But there is a wide range, so people can be affected from younger age or even very much later on in their lives, when they've already sort of had jobs and started sort of doing all the various things that you do, have children and that kind of thing. So there's a lot of variability actually even within SCA2 itself.
Got it. Yes. No, that's it is fascinating. So there's a spectrum, even with SCA2 that is typically onset at about the age of 30. There's still a spectrum of when that onset would happen. So what is there a typical patient journey on this, and like how would somebody be diagnosed with it? And if somebody was to do a genetic test, is it determinative from a genetic test that they would know that they would be getting one of these conditions at some point in their life?
Yes, so I mean, typically I think people would sort of notice some problems with their gait, with their balance and these kinds of issues. So they might go to their doctor and then get a referral to a neurologist. The neurologist would do a sort of number of neurological assessments, such as the coordination tests, or sort of looking at people's balance whilst walking. They might do MRIs. They might ask about family history, because of course, because they're dominantly inherited, there's a 50% chance of passing it on. So, you know, one of your parents would have to have had the condition. So the neurologist will do all these sorts of tests, and then they'll do genetic tests. So we've been working with some specialists and people within the UK healthcare system. So we've sort of created this pathway, recommended pathway. So it would be sort of get a referral to a neurologist. And then we've also got these specialist ataxia centres that we'd recommend people to be referred to. Because being rare diseases, a general neurologist might not come across somebody with SCAR2. So it's kind of quite useful to have people in these specialist centres where they can be supported really well by people who have seen many others with their same condition. And yes, and then in the genetic test, it would give patients the specific diagnosis of the type of scar that they have. I mean, yeah.
And is genetic testing recommended for people, or is there a hesitation for people to do it? I know in cases like, you know, with familial Alzheimer's, right? People are hesitant to get the genetic test because they just don't want to know the determinative outcome. But is this similar in ataxia, where people are hesitant to take the tests, or is it a recommended thing for people to do?
I mean, we would always say to talk to a genetic counsellor to get sort of specific advice on whether it's right for them, and you know, so talk with a neurologist, with a genetic counsellor to get sort of. But I suppose I think the majority of people, when they actually have symptoms, would like to know an answer, would get an answer. There's a lot of people who tell us that they really would like to know what they have, and then they'd be able to maybe take part in research studies. But it's not for everybody. And in terms of pre-symptomatic testing as well, if you know it's in your family, that's very much a personal decision for individuals and their families. And many people prefer not to know, and other, you know, it's very personal essentially. So yeah, from our point of view, there's no right or wrong, and we kind of advise people to talk to specialists and make their own personal decision. But I think lots of people do want to get answers. So there's quite a bit of emphasis on understanding what they actually have.
Yeah, and a lot of that usually comes down to, like, are there interventions, you know, that can help ameliorate situations, right, like this? And so with the ataxias, are there medications today? Like, what's the state of the state in terms of, you know, are there things that can slow down the progress? Are there other interventions that can support people in terms of maintaining, you know, productive and active lifestyles when they have the condition onset? So a lot of times that, you know, that impacts the thinking about how much you want to know and when, right?
Absolutely, yes. So unfortunately, there are no disease-modifying treatments, and there aren't really any treatments to sort of alleviate the ataxia symptoms per se. So the medications and treatments that there are all for any associated symptoms. So, for example, if people have the tremors or the cramps, there might be medications that help that kind of symptom, but nothing for the ataxia per se, and nothing that stops the progression getting worse. Unfortunately, that's why there really is this high unmet need to try and develop new treatments or treatments for the ataxia. So, really, in terms of the support, it's about physiotherapy, it's about occupational therapy and adaptations to the home, to mobility aids, speech therapy, and sort of support from mental health, all these kind of associated things that are really, really helpful for individuals, but not, unfortunately, something to stop the progression. Yeah, that is a big thing that we need.
Got it. Definitely, and a good reason why we're considering this project a very important one. I think it'd be helpful. We haven't touched on this yet. What is how many patients are affected worldwide by ataxias, and you know, and maybe talk about the UK as well a little bit, since that's close to home in your organization as well, in terms of, you know, number of people this addressed and the size and scope of the unmet need right now is clearly it's an important unmet need.
Yeah, I mean, epidemiological studies are lacking really, but we kind of say in the UK, we think there's about just a bit over 10,000 people with a different range of ataxias. And that's kind of just there to give us a bit of a feel, to explain that they're rare diseases, they're rare conditions, but not ultra rare when you sort of group them all together. In terms of scars, so in the UK, for example, in our registry, we have about 100 people with SCAR2, and that once again gives a little bit of a feel for numbers. Globally, I mean, in terms of the scars in general, what the papers say is a prevalence between 1 in 5 in 100,000, and SCAR2 tends to be is one of the more common ones. But there's also a lot of variation in countries. So depending on which ataxia it is, there might be, you know, pockets. So SCAR3 is very prevalent in Portugal, and SCAR2 in Cuba, and this, that kind of thing. So obviously there's differences worldwide, but it kind of, yes, and we don't have really specific numbers, but I think these numbers help to give a bit of a feel of the need and of the prevalence of the different ataxias.
Got it. Now, so that's hugely helpful on ataxia and the various types and how it affects people and what the state of interventions are and the size of the market. Now, pivoting to the research and some pathways to therapeutic interventions, how do you think about your research strategy at Ataxia UK? Like, what are you looking at? What are your priority areas, and how do you think about differentiating versus the different types of ataxias?
Yeah, we basically operate in lots of different areas within research with the overall aim of trying to move toward treatments and access of those treatments as well to people with ataxia. So we have a small funding program where we fund research projects, and we're reactive, so we invite applications through from researchers globally, and we kind of prioritize research where we're sort of where people will come to us for generating the initial pilot data, and then we might support them in trying getting funding from larger funding bodies because our grants are quite small. When we sort of look at the impact of the research that we fund, we do an annual monitoring, and because we do these small grants, it's quite satisfying to see that actually for every pound invested by us, researchers go off and get further funding for ataxia research of another £5. So it's kind of like a big shift because of our small grants. And it means that people that we're funding are staying in the ataxia field and carrying on ataxia research. Is really positive for us. So in terms of the funding program, we have this sort of open call for people to come to us, and then we have a very sort of standard but stringent way of evaluating the research with peer review and the scientific advisory committee and all of that kind of thing. And that's sort of one side of the work in research, but we also do many other sort of activities within the research field. So we will support the engagement of patients or people with ataxia in research projects. So we collaborate. Sometimes researchers come to us to be part of projects, a little bit like you're doing here today, I suppose. So you know, many EU-funded projects or sort of larger funding will come to us to get patient representatives, and we might organise workshops with people with different ataxias to get their input in study designs and priorities and that kind of thing. So we do a lot of work trying to really make sure that people with ataxia are at the centre of the research and are driving the research. So that's another area. And another thing we do very much of, we've started doing in the last few years, is research conferences for researchers and pharma companies. So we've been collaborating with other organisations, to US Ataxia Charities and Ataxia Global Initiative, and sort of creating these large conferences, which seem to be becoming more and more popular, which is great. And I think we're really sort of filling a hole that's needed to sort of promote ataxia research. And so a number of sort of recruitment to studies, because we have a registry of people with different ataxias, we can help recruit to studies and get involved in many surveys to collect data and things of that kind. So there's sort of, we do many different things, I suppose. So yeah, it's sometimes difficult to prioritize, want to do everything, but yeah.
Yeah, great. And then, so how would you characterize the state of funding for ataxia, particularly around you know, therapeutic development, translational research, new drug development? is the space in a state of being underfunded, severely underfunded, or sufficiently funded, or how do you think about that on the spectrum of, attention and amount of money that you're getting to pursue the cause?
I mean, I think, we're suffering similar problems to other rare diseases. They're not recognized, so they're not going to, they're not a priority for the big funders, for governments. So it is a struggle to get funding for the ataxias from these sort of larger bodies. And of course, in terms of charities, we've got less people to donate because it's a rarer condition. So it's sort of from all angles, really. I mean, in recent years, there has been a lot more interest from companies compared to when I started. There really was a handful, whereas now there's many more. So that's really encouraging that they're recognizing that, the research developments are leading to the point where, there's a better understanding of the conditions, of the mechanisms of action, more drugs are coming forward. So there's a lot more interest from companies developing programs. So that's really encouraging. And I think there's quite a strong ataxia academic community as well, who's sort of committed to the research. But yes, funding is always a big problem. It is always something that, we want to do things much faster and with more funds.
Good. Well, hopefully we can be helpful there, because I definitely founded Cerebrum with the idea of moving things along faster and getting more shots on goal and more diversity of hypotheses to try to advance things quicker. With the power of the people that are affected by these conditions really helping us. So Peter's been waiting around patiently, so I do want to pivot to the project at hand and talk a little bit more about our SK Channels project. It's one that fascinates me, and just from the history of Cerebrum DAO, when we were just getting started, before we really had fully developed even our community, but we just had the hypothesis of what we wanted to go build. I had done some outreach to tech transfer offices at various universities, and one of those was the University of Texas Southwestern, and they were generous enough to host me for part of the day. I shared our mission with Cerebrum DAO and what we were seeking to fund, and they curated about a half a dozen researchers from their institution that had projects that would be of interest, that were interested in talking with us about potential funding. And one of the projects that was presented was this SK Channels opportunity. And even though when I started Cerebrum DAO, the initial concept was we wanted to focus really intently on Alzheimer's disease, but then we really did expand it to being all brain disease in terms of our mission and goal. This project popped up, and it was notable in that it was research that was derived from a project that had made it to phase 2 clinical studies with a biotech before, but it seemed like it was a young biotech company that had this therapeutic opportunity that ended up getting acquired by a biotech company that was acquired by another biotech company that was ultimately acquired by big pharma for a different reason. And it seemed like this therapeutic kind of got lost in the shuffle, yet it had made it to phase 2, which gave me interest. It was like, well, that was intriguing that it would have a good safety profile likely. And the researchers came up with a patented new approach to this that they feel is at least 100 times more efficacious as well. And so that was intriguing. And so we dug in, and a year and a half later, we finally got the opportunity to work on this through a lot of good work from Peter. So it's kind of it's a great story from Cerebrum's standpoint, because it's like one of our earliest opportunities that we started pursuing. And it also is the one that took us the longest to get to the finish line, at least to get started on it, if that makes any sense. So Peter, I want to turn it over to you, and maybe you can share a little bit more about what the SK Channels project is, and what your thoughts were when it was first presented to you, and your impression of the science as well.
Yeah, thanks, Brian. I'm gonna take the audience back a little bit to telling a little bit about the SK Channels and how the SK Channels were originally promoted to be addressing the symptoms in SK2. Julie already explained, the ataxias, the predominant groups is characterized by genetic mutations. SK2 is characterized by polyglutamine repeats, which affects the Purkinje cells. That's one therapeutic option to see if you can directly intervene with these polyglutamine tracts. That has been done in other diseases as well, has not been so successful other than maybe going through the gene therapy way. But if you're looking at other options, one of the channels that is expressed throughout the body, but also specifically on the Purkinje cells, and specifically these SK2, and SK stands for small conductance calcium regulated potassium channels. And so a long sentence for a protein. And it was actually shown that if you mutate the SK2 channels, you create tremors. So the idea was that maybe these channels can play a role if you activate them in kind of stabilizing the firing pattern, which is characteristic for SK2 and many other ataxias. So it was an idea that was formed a long time ago, and there's a lot of active research overall in ion channels, specifically also on these calcium regulated channels. It's part of a larger family of ion channels. This one particularly is a smaller subfamily. It has 4 members, and SK2 is one of them. And as you just mentioned, people have been investigating the potential in actually making what is called an allosteric modulator, or positive allosteric modulator of these channels, seeing if you can actually control the firing. So people went investigating this channel, see how it was regulated, understanding the structure of the channel, and then started to kind of work on the major aspect of the regulation of this channel, which goes through calcium, as I just mentioned, which goes through a protein which is called calmodulin, which binds to the SK2 channel. And you can activate these channels also with other molecules that are interacting with a specific intracellular site on calmodulin. That has led, as you just mentioned, to academic research that has led to startup companies, et cetera, where finally Novartis acquired Cadent, as the company was called. And for one reason or the other, we will never know, shelved the project. Could be strategic, could be commercial, but as you pointed out, there is no evidence so far in the literature that tells us something about side effects of modulating this channel. So what was interesting about this project was that they took all the evidence on all the structures that were out there and asked themselves, can we actually improve on the current activities? Because if you looked at the Cadent molecule, had sufficient affinity for the SK channel, but it was not just binding to the SK2, it was also binding to another family member, the SK3. That did not introduce specific side effects, but it lowered the affinity because it's binding to other places on the Purkinje cells as well, but also throughout the body. This lowers the overall efficacy of these molecules. So the researchers, as you just mentioned, that were at Southwestern University in Chapman, asked themselves, if we look at all the structures of these molecules, and also take into account that we know now a little bit more about the structures of the SK channels itself and how calmodulin is bound to these channels, maybe by what is called structure modeling, maybe we can see how the molecules that we have now actually fit to this protein and can optimize that. That's what we are doing for the last 20, 30 years. We can model the structures of a small molecule into a larger protein structure and then see how the binding is. And then based on that, predict novel structures that may be more optimal in binding. And that was what these people had been doing. And I was quite intrigued by the quality of the research. You often see academic research touching upon something and then moving to another subject. This went really deep, and they did a lot of quality control also on the chemistry. So the chemistry was pretty good. The idea is on creating actually the selectivity on the 2.2 over the 2.3 SK channel was very well supported by data, and they actually produced then around 30 to 40 lead structures. And lo and behold, they all bound to some extent, but also 2 of them were binding actually with quite some selectivity on 2.2. And as I said, that was intriguing, and the data, also the electrophysiology data, actually correcting it in an in vitro setting on Purkinje cells was very convincing, with the additional evidence, as you just mentioned, that the project had gone all the way to phase 2, led me to believe that we had a good starting point. And also for us, which is important to build upon, we had novel structures that would not impede with the structures and the patents from Novartis. So that is the basis, and I think we were finding ourselves in a very good position with these molecules.
And then how maybe walk the audience through how we evaluated this project. So how did we look at the translational potential? And then we've decided as a first step that we wanted to replicate the research. And so I know we're in the process of doing that now. So what does that mean? Why do that? And how will that be done? And what's the timeline for that?
Yeah, there's 2 important aspects to, I would say, any project that we are evaluating. And that's, first of all, certainly of projects that we are looking at in basic research, is there evidence that this translates? How do these ion channels actually in models affect the cells or model systems, and does it reflect what we know as the clinical symptoms in SCA2 in this example? So we really were looking at, does the evidence that has been produced predominantly preclinically, does that actually translate? And as we already were saying, we knew that obviously Novartis had been taking this all the way to phase 2. So we knew that at least the drugs were safe and tolerable, that mechanistically they had shown in preclinical models all the evidence that they would be able to correct the tremor in SCA2. That was one aspect that was very important. So the translatability of the biology was there, not demonstrated finally in a clinical trial in patients, unfortunately, but all the way up there, I think the data was very strong, broadly supported in the literature. And then the chemistry, which is the other part. The molecules that had been produced were drug-like. They all showed all the characteristics of potentially becoming good drugs, although they were leads and not final drug-like molecules yet. So we were also asking ourselves, you remember, does the chemistry that we're seeing and the structures that we're seeing now provide a good route to optimize to real drug-like structures? And is there anything with regard to the structures that we can see now that we can de-risk them with regard to side effects? In other words, do we, from the structures, already recognize that there are potential red flags for future side effects that we might see later on? So we've worked with their experts in our network, and we contacted medicinal chemists who evaluated the molecules, the synthetic routes, and the potential of how they could be built to drug-like molecules. And of course, it was not black and white. It was bad or it was good. It was like a gray zone where it was clear that they had a potential of becoming a true drug with a selectivity that eventually could be tested in men. So with all these actually positive data around, and the quality of the data that was around, convinced me that we should move ahead and try this and take this further.
Now, assuming we were successful on the replication side, what would be the next steps for the research on the translation front?
Right. Yeah, as you point out, we are now looking at replicating at least the basic fundamentals of the project. That means the molecules. Can we reproduce the molecules that have been published? And do they show the same effect as has been published? That is the main aspect of this project. Once we have replicated it, basically, we will look at the different leads, and then we will build what is called a structure-activity relationship, because we have different molecules now either published or generated ourselves. And with the model at hand of the protein of the channel and the calmodulin, we are actually able to understand better how these molecules bind and why they are selective of 2.2 over 2.3. If the novel structures that we synthesize follow this principle, and we can actually prove this, we can synthesize further molecules which have better selectivity even of the ones that have been published by Southwestern University. And then it's, in principle, once you have these leads and you have proven that they're selective and in minimal setting, because we are testing them in vitro, they don't kill the cells, so they have to be at least not cytotoxic. That's a kind of a encapsulated in the experiment, some hints that the molecules don't cause any side effects. Once you have these series of molecules, you start testing them in vitro testing sets in which you are looking at, are these molecules soluble? Are they stable? We want to make sure that the molecules are not broken down in all kinds of metabolites. Metabolites is always a risk factor in drug discovery. So we are going through a number of standard tests, which is called DMPK, which looks at distribution. How does the molecule distribute in vivo? How is it metabolized? Is it metabolized? And what is the PK? In other words, the pharmacokinetics. Where will the drug go? And in this case, it will have to go to the brain. So it will have to pass the blood-brain barrier, which is another aspect on the chemistry in which we will optimize. So once we have done the replication, we will go through a series of testing and optimizations of the molecule that, as standard in drug discovery, will hopefully lead us to a molecule that fulfills the criteria, which is soluble, which shows good pharmacokinetics, which is stable, which also has a good half-life, so that when patients in the end would be treated with a drug, they would have to probably not take a pill more than once a day. That's the ideal situation, or even less maybe. So these are the aspects that we are going to work on. And as I said, it all is towards optimizing to drug-like properties that can then finally be tested in a suitable model for SK2. Initially limited, of course, to small animals, but we will increase the size of the animals to make sure that all the aspects of the drug fit with possible use in man.
Got it. And you know, for the benefit of everybody out there, at least that has tuned in, that is part of the ataxia community that wants to follow this. So on Cerebrum DAO's website, so cerebrumdao.com, our projects page, all of our projects are featured there with a fair amount of detail about the projects. And so this project, this SK Channels project, is on our website. There's a detailed project page on it, and we do publish updates, you know, to that site and through our social media channels. As we progress from the replication study, which Cerebrum has funded, we will be moving forward on the research and the translation side, and raising further funds to fund the next phase of research. And how that project gets managed, then we turn into a something that's known as an intellectual property token. And so there is the possibility for people to be able to purchase tokens in the project itself, which gives you voting rights over the project. And then we're looking to add in, through some new features we're looking to bring to our website later this year, in terms of an application where people can then get other utility and participation opportunities in the research projects as we move them forward. So these are ways that we're trying to make it so that patient communities can be active in the governance and decision making, even, and funding of specific projects. And so that's all coming soon, and will be easily available through the website. So I just want people to know that that they can at least track this project there and can participate at whatever level they want. going forward. So this is one project that we're hoping, to advance through translation. It's got some real promise we're seeing, based on history and based on the due diligence we've done in scientific assessments. I'm curious, and this will rope you back in, Julie. I'm sorry, with these calls with bridging translational development and research and patient community, sometimes we have to go a little bit long on each side. But what's the current therapeutic pipeline looking like? Are there any promising treatments or trials that are underway right now that have you excited for their potential for the community? How's what's the lay of the land right now for potential interventions for your community, and maybe Peter, a little bit on how this might fit into that and other competing technologies for what we're trying to do with SK Channels.
Yeah, I mean, for a specific type of ataxia called Friedreich's ataxia, which is not really what we're talking about today, but it's a big thing for our community. There has been the first approved drug. That was approved in 2023 in the US, and we're really pleased that actually in 2025 it was also approved here in the UK. So that's kind of a big thing for our community, and it's giving us all hope that from the first one getting an approved drug, there will be more following. For the scars in particular, the sort of the program that's kind of advanced the most is the one from the company Biohaven, using the drug troriluzole. So that's sort of something that's currently being evaluated by the regulators, and something that they're hoping will address a number of scars. Then there is the drug riluzole itself, which is the sort of troriluzole, the prodrug of riluzole. So that's another sort of drug that's been investigated. And then there's a number of sort of groups looking at antisense oligonucleotides or gene therapy kind of approaches, which of course are quite different from what we're talking about here with sort of molecules. And then in terms of other kind of treatments, there's cerebellar transcranial stimulation as a sort of symptom relief kind of treatment, and obviously there's also physiotherapy type interventions that are being explored and that kind of thing. In terms of things that are more at the preclinical stage, there's a number of sort of drug repurposing kind of projects that are looking at different approaches and different drugs, but essentially in the sort of clinical trial stage, these are the ones that I've mentioned are the ones that are most advanced in the scars, in the spinocerebellar ataxias.
Yeah, and to add to that, Julie, you mentioned troriluzole, which is now in phase 3, Biohaven, which has a history on riluzole, which has been approved in the US at least for ALS. And the interesting thing about riluzole and troriluzole is that before long, nobody really knew exactly how it worked, also not in ALS, but it hits also the SK2 channel. So that's promising. So we're seeing a company that takes troriluzole to phase 3. They must have seen positive signals, of course, in their phase 2. And the fact that, and that's for us is promising, as I was saying in the beginning, we're looking for really high affinity molecules that modulate the SK2 channel. So here we might already have some positive data in the future when it is approved. And then, of course, what we are after is an optimized molecule that hopefully is even more efficacious than troriluzole. So it is very promising direction it's going. It is very good for the patient community that there is a solution ahead. And for us, it's very promising that we might actually be able to improve on that even further. So,
yeah. No, that's interesting. And you're thinking that this drug would be potentially not just for SCAR2, but for also some other ataxias?
That's an excellent question, and this goes a little bit into the direction how we are looking at projects translationally. Is that one of the roadblocks we always have in translation is the animal models, and we know that there are suitable animal models. There's one good model for SCAR2, but it always has its limitations. So what we will be looking at and what we will have a good eye for, and that's always about good observations, is seeing if this SK channel may also play a role in other diseases. And we know that it probably plays a role in other diseases. And I think this is a matter of, once we have confidence that the molecule is good, we can test it, of course, in different models. And should we make it all the way to the clinic, then it's a matter, of course, also in your phase 2, maybe bringing in some additional patients that are not SCA2, but are slightly different and have maybe tremor, but not as a result of the polyglutamine tract. That is certainly also overall in the industry a strategy to make good observations in your clinic and seeing if you can position it in other diseases as well.
So I had one other question, then wanted to open it up. We want to leave a little bit of time for Q&A as well, see if the audience has questions they might want to come in with, and then a little bit of time more on background for Ataxia UK as well. On the clinical trial side, so assuming we do proceed through our translation path and we end up getting to a point of clinical trials, I imagine with a rare disease, recruitment's probably a challenge at times. Is that a fair statement for this field? And how is that, what's the clinical trial situation for, for this category of therapeutics, and what types of challenges are faced when therapeutic opportunities do want or are qualified to go into the clinical trial stage?
I mean, I think one of the advantages having these specialist ataxia centres, so as well as us having them in the UK, they also have them in many other countries, and a lot of the researchers are being involved in natural history studies, so a lot of the patient community is already used to being part of research by being part of these natural history studies. So I think there's a lot of desire to not just be involved in a study that's not testing a medication, and there's definitely a big desire from the patient community. We get asked a lot, what are the opportunities to take part in trials? So I think from our experience in Friedreich's ataxia in the UK, I think they managed to recruit quite easily for that. Obviously, that's a little bit more common than SCA2. But I think generally speaking, also in the US, they sort of managed to recruit for the different trials. In the UK, we've not had that many trials, so I think that's why there's also this great desire to really sort of be involved more, because there seem to be many biomarker and natural history studies, but not so much intervention studies. So I'm quite optimistic about that. I mean, we get lots of people to also reply to our surveys, which is great, because that takes time and is not, people aren't benefiting from it in the same way as they might be in a trial where they might have the opportunity of actually taking a medication that works. So I'm relatively optimistic. Obviously, it's still a rare condition. You have to put a lot of effort and all that. But yeah. I think with all our sort of patient groups helping out with recruitment in different countries, these specialist ataxia centres, I think that situation shouldn't be too difficult to overcome.
Got it. We have we have about 10 minutes. So if people have questions, they can start sending them in. I do have one already, and this one's for Julie. The question is about the state of ataxia patient registries around the globe. The question further goes on. Sometimes in cases of familial FTD, there's risk of studies being underpowered. The need for patients is larger than the actual number of patients worldwide.
Yes, I mean, it is an issue. Obviously, the trials that have taken place have been quite small in terms of numbers. They might have sort of 50, 100, those sorts of numbers. So, and so I suppose, yes, it's trying to get the outcome measures that are able to sort of manage to prove efficacy with those low numbers. I mean, the encouraging thing is, at least with the Friedreich's one, they were able to do that. I suppose, we'd have to do multi-center trials, global trials. So yeah, I mean, it's not really easy, but I think there are ways. There's all these new digital markers, there's new trial designs, there's lots of different things that can be done. I mean, even for ultra rare conditions, you can manage to get approved treatments. So if you can do it for ultra rare, you should be able to do it for the slightly less common rare conditions, I'd say.
Okay, we have a few other comments and questions, so I want to put the comment out first, and I think it may come in the form of a question. But the comment was that was speculating that the ataxia is underdiagnosed due to lack of genetic screening. Is that a fair statement to make? It seemed that could be true.
I suppose it depends on the country as well. I think there's a lot of issues with the countries because, of course, in the UK we have the National Health Service and we have the whole genome sequencing that's now been established. Although there are problems, delays in getting results and all of that kind of thing, it probably depends on the type of ataxia that you're talking about as well. Some of the more common ones are more likely to be detected sooner. Yeah, I mean, I think that there is probably an issue that there are some people that are not diagnosed yet. And that's another reason for our recommendation to go annually to see your neurologist. So as new genes are found, new genetic tests might be available. So that's an important recommendation that we make to patients, is to, even if there are no treatments, to still go and see a neurologist or specialist centre annually to sort of see if there's anything can be done to monitor or to sort of improve some of the new symptoms you're getting, but also in case there are new genetic tests available, because they're constantly finding new genes. I mean, at the latest conference, there was recently there was SCAR27B, and then before that there was RFC1. So many new genes have been discovered even in the last 2 or 3 years. So there's constant developments, and that will help with diagnosing.
We have another question coming in and related to another one. So, this person wrote, sadly, that their family suffers from SCA1. Several people have passed away, unfortunately, and sadly from it. I realize this project has a focus on SCA2, but could this be beneficial for SCA1? And then another person had asked the question regarding SCA7. So I don't know, Peter, if you want to make a comment on it, but it's sad to hear and sorry to hear that from the audience member, and just highlights the importance of funding going into projects like this and the work that we're doing. I'll let you comment on that, Peter.
Yeah, unfortunately, we have the expert in the room, Julie, with regard to the different ataxias and the different SCAs. And SCA1, of course, is distinct from SCA2. I haven't come across in our work that this particular therapy might work on SCA1. Well, maybe, Julie, you can comment on that. I think SCA1 is quite distinct from SCA2.
Yes, I mean, there are some treatments that could, so for example, the antisense oligonucleotide approach, because they're both polyglutamine conditions and you can sort of target the CAG repeats. So there is some work ongoing on those treatments that would work for both SCA1, SCA2 and SCA3. But for this particular thing, I'm not sure. For this particular drug, it probably isn't, because if those calcium channels are not involved in the same way. But there are other approaches that you can combine more than one ataxia. And the hoping is for more than one SCA as well. They're hoping to sort of get approval for all SCAs, in fact, so the trial results will work. But maybe it's something that could be explored further from Cerebrum side to see what other conditions, if any of the other ataxias, would be relevant at all.
Yeah, I mean,
yes,
that's certainly the case, and that was already brought up by Brian before. We will, of course, be looking at other indications, and there are a number of ones which you already have on the radar. As I said, SCA1, we would have to look very particular again. In this particular case, it's very much around correcting the tremors with this type of ion channel. If SCA1 pathophysiological mechanisms are different from SCA2, which I assume, it might not work, but it's something that we will further explore as we also generate the tool molecules to test in other conditions.
Yeah, and we did have a comment from one of our senior reviewers that the compounds in this project may be useful in different genetically distinct SCAs, not only SCA2. So that was one of the things that made that person in particular very excited about this project. So about its reach and potential. We had another comment, Peter. This is interesting. Came from the community. It said they looked at our website and noticed that our project Transfidelity would also have potential benefit for ataxia. I don't know if we've given that any thought around translational fidelity and ataxia, but it's something to take into consideration.
Well, we haven't particularly given that consideration for SCA2 because the idea of transfidelity, of course, is about aging, about neurodegeneration, and about the translational machinery that translates from mRNA into proteins gets faulty over time, and that leads then to a number of wide range of neurodegenerative diseases or other aspects of aging. So as this is a very distinct group of diseases with a very distinct let's say causation, I don't immediately see a relation to transfidelity and ataxias, but yeah.
Think about that. Okay, I have one last question, and we've got about 3 minutes left, so I want to give Julie a chance to close as well beyond that. But question for either one of you. I think this will go to Julie. Could you discuss the availability of biomarkers for clinical trials? Is there anything beyond the clinical rating for ataxias?
Yes, so there's quite a lot of work going on trying to develop new biomarkers. There's the Ataxia Global Initiative, which is a sort of network of ataxia specialists who are specifically looking at trying to get to the point of trial readiness. So there's sort of fluid biomarkers for the blood, like something called NFL, that's showing promise in a number of neurodegenerative conditions, including the ataxias, and papers on that. So that's something that could be used as an additional way of seeing whether a medication is having a positive effect. There's a number of sort of more digital wearable devices that are being used to measure gait, to measure coordination. There's sort of there's a group of researchers who are working on something where you sort of use a spoon, an instrumented spoon, that then you can get measurements. So there's lots of other different biomarkers that are being developed so that you're not just relying on the clinical rating scales, as well as some patient-reported outcome measures as well, where you're sort of asking patients how they're feeling in lots of different quality of life aspects. But it is a very active field, so there's still a lot going on in that, and there's a lot of desire from the ataxia community to do more work on that, to develop the best ways of measuring the effect of medications in trials.
Okay, great. Well, we are coming up to the top of the hour. I want to thank you both for such a thorough and informative discussion on what's clearly a very, very important topic. Julie, is there anything else you want to share about Ataxia UK, about your organization, how people can participate and support you before we close?
Yes, sure. I mean, if anybody who's on the line is affected by ataxia, please do join Ataxia UK. It's free to join. We provide lots of information, opportunities to take part in research, as we mentioned, but also information, stories. We share stories from people with ataxia about how they live with the condition, sort of empower people. There's a helpline. There's all sorts of bits and pieces of information and things that we try and do to support the community. So do feel free to join Ataxia UK if you're interested, to support our work and to be supported by us. We have a virtual annual conference in October, so that will be a sort of useful event for everybody, hopefully. So and I can see that the website's on the screen. So yeah, I think that's just sort of to sum up. We're here for anyone who has ataxia.
Perfect. Well, thank you both for your time and for the work that you're doing for this community, and thank you to the community members that came from both Ataxia UK and Cerebrum, and from that found us through the internet too. We're all glad that you're here. Definitely follow along on this project, SK Channels. We welcome your input. We welcome your participation, and we hope that we have something that's promising on the horizon here for this community. Thanks again, everybody, and we're going to wrap.
Thanks.